So I think you are very much clear about the structure of a mature hermaphrodite proglottid. So it is having both the male and the female reproductive organs. So it has got numerous testes, whereas it has got only one ovary, which is bilobed. So we had an oviduct, we had an ootype, the round chamber, and then we had the uterine canal, the uterus. It had the two parts. One was the narrow part, which we call it as the uterine canal, and the second part was the broader part, which was the extended part of the uterus. Then we had the vitelline gland, which produced yolk, and we had the vitelline duct, which again was joining the ootype. And the third thing which joined the ootype was the oviduct. So the vaginal duct, it was joining the oviduct. So what will be the physiology? That first of all, what happens, the sperms, they are carried from the vaginal duct up to the oviduct. So if we go by the process of the fertilization, here the sperms, they are entered. So the cirrus of some other proglottid, it will inject the sperms into the vaginal duct. So these sperms will be carried down from this vaginal duct up to the oviduct. So now the oviduct, it is carrying the ova from both the sides, and these ova, they are coming down, and in the oviduct we have the process of fertilization. So in the oviduct itself, the fertilization occurs. So this is the place where fertilization occurs. Now this fertilization is a cross type of fertilization, because the sperms of this particular segment, they are not fertilizing the eggs of this segment. So the sperms, they are coming from some other source. So that's why we call it as a cross type of fertilization. Now students, after fertilization, the eggs, or you can say the zygotes, here we should say zygotes, they are entering into the ootype. So after fertilization, the eggs, they come down, or the zygotes, they come down into the ootype. So when they come down into the ootype, what happens? There is a special gland which is called as the Mahley's gland. Now this is a very, very important gland as per the function is concerned. Now this Mahley's gland, it is producing its own secretion, which is going in for multiple functions. So Mahley's gland secretion is functioning to lubricate the passage of the eggs. And one thing which is to be noted, that at the lower side we have the vitelline gland. So this is the vitelline gland, and it is producing the yolk. So what happens, that both the yolk and the Mahley's gland secretion, these are poured over the zygotes. So this is a very important kind of mechanism. So if you go with this particular part of the diagram, so what happened till now is that the sperms, they were coming from the vaginal duct, they were coming down up to the oviduct, and ova, they were coming down from the oviduct as such. So in the oviduct, the fertilization occurred, and then the fertilized eggs, they came into the ootype. The vitelline gland secretion is again coming into the ootype. And third, the secretion of the Mehli's gland, it is poured onto the zygotes. So here what happens, that the ovum is surrounded by now the yolk cells. The upper yolk cells, they form a particular membrane which is called as chorionic membrane. And when this chorionic membrane is formed, now the zygote is called as a capsule. So that becomes an egg capsule. Its outer layer, it hardens and it becomes capsule. Now this capsule, it goes up into the uterus in this direction. So after the formation of the egg capsules, they go to the narrow part of the uterus, and after going into this narrow part, they go to the broader part and they are stored over here. So here you can very well see that we have the egg capsules in the uterus. So they are stored. So this is a mechanism of the formation of the egg capsules. Now how they are formed. So the uterus, it may have 30 to 40,000 egg capsules in a single proglottid. Now this particular uterus, which is in a single segment, can have 30 to 40,000 capsules which are stored in this particular proglottid. So you can very well imagine that how much number of egg capsules is produced in a TAENIA solium. So it has got, suppose it has got 300 mature proglottids, out of which 200 are like hermaphrodite matures. So 200 multiplied by 30,000. So this is the number of the egg capsules that it can produce for its next generation. So when the eggs or the egg capsules, they are passing from the ootype into the uterus, the secretion of the Mehli's gland, it helps them to lubricate the path. So it helps in the easy passage of these egg capsules into the uterus. So that was the physiology of the reproduction, or you can say the formation of the zygote or the fertilization. And this is the mechanism of the formation of the egg capsules in case of the mature hermaphrodite proglottid. So students, now in this part of the video we have discussed the male and the female reproductive systems of the TAENIA. We have discussed the physiology, that how the egg capsules, they are formed, and now you know that the uterus has got the stored egg capsules in which the zygotes are present. Now in the very last part of the TAENIA, we will discuss and we will start from that zygote, that how a TAENIA is developed and what is the life cycle of the TAENIA solium. So up till now you should remember that how the egg capsules are formed, and now they are stored in the uterus of the mature proglottids. This particular slide is showing you 3 mature proglottids. Now, if you see these proglottids carefully, you can very well see the presence of the ovary. So you can see the bilobed ovary. These are the 2 lobes of the ovary, and you can very well see the presence of the vitelline gland. So this is the lower vitelline gland present, and if you clearly see, if you see it carefully, you can very well see this part is the O type in the very center. So this is the O type, and here we have the vas deferens. So some of the things are clear in these mature proglottids. And one thing I had told you in the very last part of the lecture, that we will discuss the genital papillae later on. I told you. So these are the genital papillae. So this is the genital papilla. This is the genital papilla. So these genital papillae, they are alternate to each other. So if you go by this papilla, this is on the left side and this is on the right side. So next will be on again the left, then we have on the right, then left, then right. So they are alternate in position in the taenia solium. So these genital papillae, they are having both the male and the female reproductive tracts opening into the genital atrium. So genital atrium was the depression in which both the male and the female gonopores were opening, and genital papilla, it is having the genital atrium inside it. So that was about the genital papillae. They are alternate in position, and they are only and only present in the mature proglottids. They are absent in immature ones, though they are present in old proglottids also, but they are non-functional over there. Now students, the next slide is of the gravid proglottid. So now we have to discuss the structure of one old proglottid. Now if you see the actual structure, this is a microscopic photograph of a gravid proglottid. So here you can very well see the presence of some branches, or you can say branched uterus. So all these branches are of uterus. So this is very important to note. The gravid proglottid or the old proglottid has only and only one thing left, that is the uterus. The testes, all the testes, the ovary, the vitelline gland, everything is diminished. Everything fades out. So only and only uterus is left in the gravid proglottid, and you know that uterus has the stored egg capsules in it. So this branched uterus is having only and only the egg capsules stored in it. So this is the most important thing which you should remember, that if you have been asked that what the gravid proglottid has got, so it has got the stored egg capsules. And why it is branched? Why the uterus is branched? So the answer to this question is that to store more of the eggs, more of the egg capsules, the uterus, the broad part of the uterus is branched into many branches, so it can store a large number of eggs or the egg capsules. So if we go by the structure the diagrammatic structure of the gravid proglottid. Number A, the A is the branched uterus. So you can see number of branches of the uterus. So the branching is because it can store large number of capsules. Testes are degenerated, ovary is degenerated, and only the genital papilla, B is the genital papilla. I have earlier told you that genital papilla is non-functional in this proglottid. Now the C part, this is the remnant of the vas deferens which was present in the mature proglottid. So this is a remnant of the vas deferens. So you may have the serous in gravid, the gonopore, we have the sperm duct, we have some of the vaginal duct. So these are the remnants present in the gravid proglottid, and all these are non-functional in nature. So what is functional? It is only the uterus, because it is containing egg capsules. So this is the structure of a gravid proglottid. Now students, there is a process which is involved with the development of the taenia solium, and this is called as apolysis. So apolysis is a very important process which is present in taenia solium. Now, as we know that the word lysis means the breakdown. So this is breaking down of something in taenia solium. So students, we know that the taenia solium is, you can say, it can reach up to 5 meters length. Now suppose this is the scolex. I am trying to draw one taenia solium. Then it has a small neck, and this is the body of the taenia, and now it has got the 5 meter length, suppose. Now what will happen that if it grows more than 5 meters, suppose it is growing more than 5 meters. Now this is the overgrown length, which is shown here in red color. Now this overgrown length will be cut down. Now this length will be cut down in groups. Suppose this is the first group of proglottids. So you must know this, that the end segments will be containing the old proglottids. So apolysis is the breakdown of the old proglottids in groups from the body of the taenia solium. So what will happen that the last proglottids, the last old proglottids will be cut off, and they will be thrown out of the body of the taenia. So what will happen that if we take taenia just like this, so if this is the body of the taenia and these are the cut off proglottids which are broken down in groups. So this process of breaking down of the old proglottids is called as apolysis. Now why apolysis is important? This is the foremost important question. Now what happens? First of all, we have to see that where the taenia solium is present. So taenia solium is present in the intestine of man. Now suppose this is the digestive system. If this is the mouth, and we have the esophagus, then we have the stomach, then we have the small intestine, this is the large intestine, and this is the outpore or the anus. So where we have the Taenia solium, it is present in the intestine. Now if some of the proglottids, they are broken down, suppose these are the last 20 proglottids which are broken down. So where they will go? They will be going through the large intestine and they will be going out of the body. That means the apolysis happens, it breaks the last proglottids, and those proglottids will come out of the body of the man with the feces. And the most important thing is that what is present in those gravid proglottids. So if these proglottids, they break down and they come out, they have the egg capsules. One gravid proglottid may have 30 to 40,000 egg capsules. That means if you are releasing 10 gravid proglottids, that means you have released near about 3 lakhs or 4 lakh of the egg capsules into the environment. So this is the importance of apolysis. This is a kind of dissemination of the egg capsules into the environment by the Taenia solium. So you should know about apolysis, that this is the breakdown of the last old proglottids of Taenia in groups.